Hands up, so many. So I'm going to talk about the challenges and opportunities. So growing our knowledge. By attending these seminars, by going, and if you are not updating your knowledge, then you are out of the market. They brought a revolution with Android. So this is a great lesson for us. If you are not updating yourself, you are nowhere in the market. You will be thrown out of the market. Always try to see that how you can update the knowledge, how you can be along with the market. If not leading, at least along with the market. So always try to update your knowledge, try to attend these seminars, try to attend different conferences, and keep on updating. It is a continuous learning. Even at this stage, after 20 years, the more I learn, I feel that still I don't know anything. Still there is something to learn. There is a continuous need of learning for us. So today I will try to show you some of the consideration when you do research, especially for the infectious diseases. Please see this slide.
Yes, sir. Yes.
The contents of my slide is just I will highlight about the history of infectious diseases, and next we'll see about the consideration of infectious disease research, what you need to consider. There are multiple factors you need to consider. At the same time, we will be learning about the challenges and opportunities in infectious disease research. And finally, I will conclude the session with the last one slide. So when we talk about the history of infectious diseases, this is nothing new for us. Since the dawn of human beings, we have been seeing some kind of diseases. And of course, there were different phases. There was different mode of medications which was used to treat the diseases. Even before identifying or discovering the bacteria or pathogens or microorganisms, there was some way to deal with these organisms, to deal with these infectious diseases. And basically, for infectious diseases phases, they are divided into 5 different phases. Starting with the 15th century, you are having a classical medication where Hippocrates and Galen, they were leading the medical field and they were trying to treat the patient. At that time, even they doesn't know what is bacteria, they doesn't know what is virus, but still they were able to success to treat certain diseases. But still there was more to learn. There was second phase, 16th to 18th century. There they started the traditional system of medication. They started giving some herbal remedies. Even during that stage, they didn't discover the pathogens, but still they were treating it. Still we have many things to learn. There was some development. 19th century, you can see the early modern medication. They have discovered the bacteria. They discovered certain vaccines to treat these infectious diseases, to tackle with these infectious diseases. And next phase is about 20th century. We can say a late modern medication. We have seen the discovery of penicillin, the discovery of antibiotics, the last production of antibiotics. Of course, it has treated a lot of infectious diseases, but still there were many challenges. Still there is a lot of things to learn. And now you can see we are in the 21st century. But still, you have seen, you are evident that they are not able to handle the first pandemic, the coronavirus. They are not able to handle it. Although we have more technologies, there are new technologies for the discovery of vaccines, for the discovery of new molecules. But still, they were not able to tackle that in the first pandemic. And in the history, it was the first time the clinical trials has entered each and everyone's home. This is the first time in history that they have tested the drug directly on human beings without any clinical trial. So this shows that still we need to learn more. There are a lot of lacunas. Although we have different technologies, new technologies, we are using in silico methods, we are using in vivo methods, we are using different methods, conjugate docking method, simulation method, but still they could not able to know the complete information about the pathogens. Of course, there are some two different classifications I can say. One is controllable, another is uncontrollable option. So there are some uncontrollable challenges or difficulties which we can able to control if we have a proper coordination. And of course, the second thing is about the controllable features, where it's not in our hands. One of the examples is about the resistance. It is not in our hand. The changes in the strains, we have seen the first attack, the second attack of coronavirus, the third attack. Even now, you can see throughout the world, there is a continuous emergence of new strains of coronavirus, which is uncontrollable. Every year we have been listening from 2019, every year we have been listening, there is a new strain in the market. Every year there is some kind of fear among the public. It shows that still we need to learn. Still we need to develop the way we are doing the research. Still there is a need to upgrade our technologies. Although we are in the 20th millennium, but still we need some higher technologies. So we will be finding out that what are the different factors which we have to know when we are doing the infectious disease research. So before discussing those considerations, I like to show about the different models which are used for infectious diseases. Like as I mentioned, they have in silico virtual screening, or they have in silico molecular dynamic simulations, or in vitro biochemical and cell assays, and in vivo animal assays. These are the different parameters they use for infectious diseases. And when we talk about infectious disease, it is not just one disease. It is not just one pathogen. There are different pathogens. And within the pathogens, there are different strains. They have the ability to come up with a different strains. So we need to know about these challenges, how to tackle these challenges. We cannot use only one model. Even if we talk about the animal model, just one animal model is not sufficient for the infectious diseases. There are different considerations when you select the animal model. In general, we can say about, while selecting the animals, we have to know about the homology, we have to know about the analogy, we have to know about the fidelity. What this terminology is. When you are using these animals, you have to see that it should have the structural similarities. Similarities with the humans, because we are using these medications for the humans. So when you are testing any animals, you have to see that there are some structural similarities in the animals. We have to select these animals based on the functional similarities. We have to select these animals which are closely resembles the human beings. We have to use the animals where there is a reproducibility. So there are different considerations in selection of the animals. What we say as homology, analogy, fidelity. And sometimes when you select the animals which have close resemblance to human beings, and again there will be some ethical consideration. You will not get the ethical approach. For example, chimpanzees. You cannot take chimpanzees for each and every experiment, although they are closely resembled. And you cannot select the animals, although they will be the best animal models, but these pathogens will not affect the animals. So there are different factors which have to be considered when you are doing the research for infectious diseases. In some you are using one model, in some you are using multiple models, and even selection of the models you have to select it carefully. We'll see the consideration one by one. In consideration infectious diseases, the first thing is about your objectives. Change the slide. Defining the research objectives. This is very much important. Why you are doing this research? What is the purpose of your research? Based on that, you have to select the animal models. For example, if you want to check or evaluate the inter-host transmission, then selecting Dunia pigs is the best model. If you are trying to check the immune response, then the rodents are the best animal models. So we have to know about these objectives. What is your objective? It is just not to treat these diseases. But the objectives are different for every scientist, every researcher. They have their different objectives. So based on their objectives, we have to select the animals. So we need to have a thorough knowledge of infectious diseases, the pathogens, and the animal models. And accordingly, whatever the objective you have, accordingly you have to select the animal. The second parameter is about the response to the infection. To know the response to the infection, first you have to read about the disease factors, the pathophysiological changes. How do you know that the person is having the infection? So when you are using an animal model, you have to identify those symptoms, progression of the disease. Other, if there is any change in the strain of coronavirus, able to taste, you cannot able to running nose, they have mixed influenza symptoms and uninfluenza. The pathogens are in the cell culture and okay. You have to know your scale or status of. So you will be having, and during the treatment, because of these bedding, the material what you are using for the bedding, what we call as extrinsic factor, it will affect the intrinsic factor of the animal, and it will affect the disease progression in the animal models. Maybe because of the leftover phases, and again it will increase the clinical pathogenicity in the animal model. Even we have to see about the hygienic condition during the research. So these are the simple factors which we need to consider. Even when you are using bedding, we have to know that what kind of material should be used so that there will not be any double infection from that. We are treating one infection in the animal, and because of these actions which are very dangerous, we can say, so where we use limited areas so that the infection should not be transferred in other areas, in other rooms or whatever it may be. Highly transmissible. So in that case, if you not use large animals within the limited space, you have to use the small animals. Otherwise, it will be overcrowded. And again, there are chances of secondary infections. If you look at animal size, what it affects them? But it will cause, because of the overcrowding, it will cause secondary infection. So again, we need to have the knowledge about what kind of animals you have to use. Whether you have to use the smaller animals, you have to use the bigger animals, or you have to use the cells. And that too we have to decide according to the infectious diseases for which parasites, which pathogens, which organisms you are trying to do the research. Simply you cannot select any animal and do the infectious disease research. is about the anatomic interactions. We need to have complete knowledge, complete anatomy of the animals before doing the infectious diseases. Without that, again, you may end up with the failure because of inappropriate knowledge of anatomy of these animals. And next we have about the personal safety. Not only hygiene for the cages, even for the personal safety. It is very dangerous to handle the infectious agents. So even the workers who are working, they will be very much prone to the secondary infections or infections from those pathogens. And organisms, virus, bacteria, whatever it may be, it is from the clinical source. It means that, and if it is from the lab sources, there are chances of attenuation of these pathogens. If it is continuously grown in artificial media, definitely the organism will be attenuated. Again, you will not get the proper results. So these are the different factors you will come across when you are dealing with infectious disease research. Other consideration is about the infectious agents, that what I said about the source, from where it comes. First thing is about the personal hygiene, how you maintain these organisms. Every lab doesn't have the facility to maintain these organisms. Every institute, they do not have the facility or safety profile to maintain the cancer cells. Every lab, every institute doesn't have the facility to maintain the viruses. One thing is that the government will not allow it. Because if they allow every institute to culture these viruses, then there are chances of different strains, again development of different strains. So it is the pure cultures are maintained by the government lab. So selection of infectious agents is another challenging or consideration for our research, infectious disease research. Last but not least, the chemicals what we are using. The chemicals what you use for the research. Again, this is challenging for you. As of now, the chemicals what you have, it might be sufficient for the rodent research. The same chemical reagents you cannot use for different other animal models. As I said that you have to use different animal models based on the different infectious disease. Majority they are using the rodent, but if they are having a different model like ferrets, they use the ferret or they use the non-human purine. So for that we don't have at the moment we don't have those kind of reagents which can be used for the other animals. Because there are a number of factors. I hope in the morning session Professor Kaviraj he has mentioned about those factors. So all these reagents will not be suitable for every animal model. So this is again the consideration when you are starting the or initiating any infectious disease models. You have to know about these reagents, whether it is available or not, whether we have sufficient facilities or not. So these are the different considerations you have to look when you are doing infectious disease research. Okay. And next, we will be discussing about the challenges. Of course, during these considerations, we have seen all these are the challenges. Again, there are some specific challenges. The most common challenge is about the resistance. The development of resistance is a major challenge in infectious disease. Before the drug comes into the market, the drug will be outdated, or the drug will be insufficient to treat the disease. This is a major challenge. Although we have in silico models, we have targeted drug delivery systems, but still it is not in our hands. The development of new strains, development of resistance, this is a major challenge. And here, every field we need to work closely to have a better outcome of infectious diseases model. We should not limit our research within our department. As of now, we have seen pharmaceutics people, they do the research, they are keeping in their lab. Chemistry people, they are keeping in their lab. Herbal medication, it is just in the lab. It is not moving out, or they are not collaborating with all the departments. Now there is a need to have a better collaboration with each and every department. We need to have a collaboration with the chemistry department, collaboration with pharmacology, collaboration with herbal medication, pharmacology, pharmaceutics, clinical pharmacy. They all should work closely so that they can have a better information about these diseases, and especially when we talk about the resistance, there is a need of awareness among the general public. Nowadays, before the drug is taken by the human being, so it will be resistant to that person. He has never taken that antibiotic, but still it will be a resistant, that the organisms will be resistant to that antibiotic. Again, there are different factors. I correlate with the research. Even though it is a clinical point of view, mostly it is required for the clinical research. I hope I was audible during those times. So regarding the awareness among the general public, as I said that a person who never taken antibiotic is developing the resistance. What is the reason for it? The main reason is the fertilizers. The main reason is the poultry food what we are eating. Whether you eat any non-vegetarian, whether it is chicken or mutton, whatever it is, they are given antibiotic food. The food which is given, the pellets what they are giving to these animals are mixed with antibiotics. So when you are eating the chicken, when you are eating the fertilizer food or whatever it may be, whether it is a vegetarian or plant-based or animal-based food, they are mixed with antibiotics. That's the reason that when you are eating this food, cells are developing resistance. The antibiotics are the third generation. If you are coming, these in the molecules. So the human beings, what we call as naturals, that when you are specific for the based on the objectives, based response. Again, I have some identify the target. Even if it is very during the disease, eradication of the organism, end up with the wrong disease, a complete cure of the disease. And after few, if you doesn't have, but if you keep on continuing the disease. For example, we can see in malaria, Plasmodium vivax and Plasmodium ovis, it can remain in the dormant state for several weeks or several years. With a new organism, it has a similar features, infectious agents. The complete into the cytoplasm. So again, as I said that when you have one, they cannot manage it. The selection of animals. So all the challenges you may come across with infectious disease models. Please next slide. I covered this resistance. Already I have covered. The last, in conclusion, of course I didn't cover everything. So why there is a why we are not able to know complete agent? new virus or new. We doesn't know why it is changing. So there is a need and the less we know to come for the development. And when you are selecting any animal models, simply try to have reasons have a better outcome in infectious disease. With this, I conclude my today's session. Always try to have incomplete knowledge. I take advantage of the expert opinions. Try to collaborate the research with different departments. Even journals now they are trying to see that whether there is any collaboration with different departments to have a better activities for in infection.
Yes, delegates, the session is open for you.